What I want to do in this video is I want to talk about the endosymbiotic theory, which is sort of the theory of how the cells of multicellular organisms came to be. But the reason I really like talking about it is that it helps us to understand how different cells are different and why. So when we go back about 3.6 billion years or so, the only cells around look like this bacteria cell. These cells are much smaller than animal or plant cells, and they have some important differences. As you look at this bacterial cell, it still has DNA. It still needs genetic material to make the stuff that it uses. But this genetic material is not held within a nucleus. Instead, it's sort of hanging out in this region called the nucleoid, and it's able to float in the cytoplasm. They're freely floating ribosomes as well, and those ribosomes have access to this DNA. They can transcribe that DNA anytime they want. Now, surrounding this cell is still a cell membrane because it needs to keep its insides from its outsides, and that's an important thing. And when we think about the bacteria very specifically, bacteria need extra protection from the environment around them. This means that they have an additional cell wall and sometimes a cell capsule as well. You may be wondering why bacteria need this extra protection, and really, if you think about it, they're found in some very difficult environments, and these protective layers will keep stuff from getting into them that shouldn't, and it may protect them from things like dehydration. Now, if you're a bacterium, you might not want to stay in one place all the time. And so one thing you'll notice as we look at this bacterium is that it's got all this sort of stuff hanging off of it that our animal cell that we looked at last time didn't. The big tail here, this is called a flagellum. And the bacteria will use this flagellum to be able to move around its environment. And if we look closer at this flagellum, there are actually little molecular motors in here that are used to cause that flagellum to beat, and those run on ATP. All right, as we are continuing to look at our bacteria, you'll notice that there are sort of long hairs and short hairs. These short hairs are called fimbria, and these fimbria are used to be able to sort of stick to things and crawl along them, and that's something that the bacteria likes to do. In addition to these fimbria, the bacteria have pili. These are the longer guys. And these pili can be used also for sort of crawling along things, but a big function of theirs is actually in bacteria being able to stick to others and communicate, and this happens when bacteria reproduce. Now, you might wonder how bacteria reproduce. Well, in addition to the DNA that they have here, they also have these little circular pieces of DNA. These are called plasmids. And the bacteria use these plasmids and basically exchange the plasmids with another bacterium. These plasmids can carry with them things like antibiotic resistance. And that's actually a really good thing. So this is how the bacteria can pass their genetic material on to other bacteria. So now we've got something that can make what it needs to survive as a cell. It can reproduce by passing some of its traits along, and it can move around its environment freely, and it's protected a little bit from the hazards of the environment. Okay, so I added a little chart here, and what I want to show you is that when we look at the bacteria and we think about the genetic material, bacteria have DNA, and then they also have those plasmids. And those plasmids are a little more stable, and they don't have to be transcribed with the rest of the genome. So they're kind of cool because they can give the bacteria additional functions without all the muss and fuss. In order to do that, bacteria also need ribosomes, which they have. They, when we think about their protection from the outer world, they have their cell membrane, but they also have a cell wall and a capsule. And when we think about their ability to move, they have a flagellum, they have pili, and they have the fimbriae. You may notice here that they don't have any other organelles, and that's a prokaryote thing. They don't have membrane-bound organelles. Now, a very natural question is, how did organelles come to be? Well, some bacteria figured out ways to create energy. There were aerobic bacteria. They were able to use oxygen to create energy. And there were cyanobacteria, which are photosynthetic. They were able to use sunlight to create energy. And the thought is that there was a third type of bacteria, and these ate the others to get their energy. At one point or another, in eating another bacterium, so if the eater is eating an aerobic bacterium, it ate it, but it didn't fully digest it. And that bacterium that didn't get fully digested was able to create energy still, and was able to create energy for that bacterium. And this is thought to be the first mitochondrion. Similarly, if the eater ate a photosynthetic bacterium, that would create their energy, and this was considered the first chloroplast. This whole idea may seem really far-fetched to you, but when we look at a mitochondrion, it looks like a bacterium. And when we look at a chloroplast, it looks like a bacterium. These structures have their own internal ribosomes, they have their own DNA, and they reproduce like bacteria. So there's a lot of evidence suggesting that they come from a bacterial lineage. So what comes next? Well, there's a need to now protect assets. And in this case, what happened is the cell put a membrane around its DNA to protect its DNA from getting damaged somehow. When it did this, it separated the DNA from the ribosomes, so the ribosomes couldn't transcribe the DNA whenever they wanted. And this kind of led to the creation of a more linear shape of DNA, which is what we're used to, rather than the more circular shape that bacteria have. These cells continued to grow, and as they grew, now a cytoskeleton was needed to move things around within the cell. And so both plant cells and animal cells are going to have these cytoskeletons. And this is used for motor proteins to be able to move stuff to the far reaches. In addition, pockets of membrane kind of continued to fold inward. And as they folded inward, they started to create more specialized structures. This led to the formation of things like lysosomes and peroxisomes, which are filled with enzymes. And those enzymes were then able to break down structures within the cell. Now, in addition to this, now that we've got all of this stuff together, we need sort of a way to make those enzymes, right? And making those enzymes happens in the endoplasmic reticulum. The endoplasmic reticulum is actually involved in folding them as well. Because everything was surrounded by membrane, now we needed a way to make more membrane, and that led to the formation of the smooth endoplasmic reticulum. So at this point, we've got a bunch more structures cropping up as we're looking at our different cells. Now, just because we have these evolutionary things occurring at the same time doesn't mean that they happen in the exact same way. So when we look at the divergence between plant and animal cells, one of the things that we see is that the plant cells sometimes have an additional cell wall, and that's similar to the bacteria. And they use that to protect them from their environment. Why? Plants can't move around, so they can't exactly move out of a harsh environment and into a new environment. These cell walls are basically cellulose, which is a long chain carbohydrate, and it's really strong, so it gives the plant some structure as well. If you look at plant cells in a stem, they're going to look like bricks, and this is largely because of that cell wall. Because you've got this cell wall protecting you and you're now in a multicellular structure, you have to have some way of communicating information between cells. This is where these little pores come in. These are called plasmodesmata, and they allow signals to flow from one cell to another. Beyond this, that cell wall does help the cell to avoid some water loss. However, plant cells or cells in general are basically big bags of water, and if you're in changing environmental conditions, you kind of need to deal with that. So there's this huge structure in the center of the plant cell. This is called a central vacuole. And that central vacuole is able to gain and lose water, and that helps plant cells to maintain what's called turgor pressure. This is what keeps that cell wall from shrinking. Now, in addition, when we look at these cells, now they're getting more complex. And one of the things that is needed with this complexity is the ability to target things when you're trying to ship them. So you'll notice this plant cell still has a Golgi apparatus. That's the plant pack and ship center. However, the Golgi apparatus has another function in plant cells. The plant cell needs the cell wall, and the cell wall is created in part by the Golgi apparatus. So it's able to process carbohydrates into cell wall material. And since it already has a sort of an in on the shipping industry, it makes it very easy for it to package that cell wall and move it around.